So, dear Professor Marquette, that's a real pleasure for me to hear you. And I will put your slides. Yes, I put your slides. I'm sorry. I have put them. Where is it? No, that's. Oh. So here and now we got it, and I put it in the. How to do that?
No,
yes, full screen. I have to put it in full screen, I think. So that's okay.
Nope.
I think now.
No, you have to go there.
Where?
This there. There you go.
Why?
But you have to switch.
That's complicated.
Oh, shit, this thing is.
Also, yeah, also at CMM.
What is the switch?
Oh, there you go. That's it? Okay, so you have it.
And now I. That should be fine. Yeah, just leave it there. Because what is
recorded? I tell you because I have it here.
Okay. I think that we should do it again.
No, it's not good yet. It is not. You
Yeah,
see it this way. So I don't know. I think you did it. No, but
Okay, there is.
You are a bad person.
Oh, wait.
Why don't you?
Wanna,
This is something else. This is.
I want to get rid of this because there is something that we can push in there behind the bar.
Yes, but if you put it in the full screen, I think it's okay.
Okay.
Wait. What is this story?
Oh, let me
wait, because here it should be.
But we have to get out of this mode. Let's get out of this mode. So you have to press now.
Yes, okay. So now what is here? So we have to go. What is the story? So afficher. So we are in the full screen mode. But yes, but now you have to. Yes, okay. I see. Yes, I understand. How to put it? I don't know. I never used this. How to? You say that there is something here.
Yeah, but because we have to switch, so you have to
wait. Maybe we stop
here.
We stop sharing. And
wait. We have to go here.
Yes, I know.
Here, and you press this switch in this screen.
Okay. We have to go where? So wait. What I do is I stop sharing.
Look. No, you move. Wait. There you go. No. Right. Perfect. That's it. Now they are. This is big here, and I'm seeing this. This is what I'm supposed to see.
Okay. And so now are we good?
We are good.
Okay. So I'm sorry for the delay. So dear Pablo, that's a pleasure to hear you, and I hope it will be a pleasure.
I hope the same too.
I know it. And so thank you for coming. Hey.
We did it.
So Pablo is in, and so I don't know if it works, but if it doesn't work,
Okay. Thank you, Paul. Thank you for the invitation. I'll be trying to talk a little bit about the main topic, at least the name of the workshop was network reconstruction. I will touch on it. I'm just going to try to make an introduction that will make more sense as you see Daniel and Mara later, who are going to be talking on kind of the same problems. So, and I'm gonna do that, and then I'm gonna talk a little bit about something that I thought might be interesting also for this audience, considering what I've been seeing in the morning, which I really like it. It was really great talks. So I hope you enjoyed this one too. So, well, the thing with networks in ecology, in biology, actually, is you might know or suspect, is a big fad. So everyone's working in networks these days, and because you can actually say very this is rebelling against me, but I have it. There you go. No problem. Yeah. So because I mean everything in living organisms is about network. So you can represent everything pretty much as a network of where you have couple basic functions like acquisition, transformation, storage, and transmission of this energy, matter, and information. So you find this across every level of organization, that these processes are happening. And you can actually try to understand this by constructing a network of interacting entities that are involved in some of these processes. So, and as you might know, this network theory has become big with the work of Strogatz, Barabasi, and some other people. And now, you know, it's a way of describing ecosystems like food webs, like here, or grid systems, metabolic systems, computer systems, everything can be understood as a network. So what I'm going to talk a little bit is this network of a graph with these vertices and edges. And I'm going to make the point that there is fundamental uncertainty in terms of the nodes or the vertices in this network, in terms of how many they are, how they change, and also fundamental uncertainty on the linkages and how perturbations are propagated through the network while you move something here, you know, especially if you start adding things or removing things, what we call a press experiment. Suppose a press experiment when you have a system and you start, you continually press on it, you know, by suppose spilling oil or including a pollutant in the system, or by putting greenhouse gases like now, and the system start changing. So all of those continuous press experiments are important, and there are some results that say that the way causality propagates through the network is impossible to actually ascertain with certainty. So it's indeterminate, you know, which is very interesting because we are in the middle of a huge experiment at the planetary scale. So we need to be there and understand. So in ecology, the prevailing view back in the 60s can be expressed, for example, in what MacArthur said, pretty much that diversity begets stability. You know, we were talking in the morning about the portfolio effect, and the portfolio effect usually convey that feeling that the more diverse the system, the more they can, you know, buffer each other in the fluctuations, and you get a more stable system. So that was the prevailing view, and MacArthur was one of the champions of that view until someone published a note, a one-page note in the journal Nature, and that someone was Mark Gardner and Ross Ashby. Maybe you know Ashby because Ashby is one of the fathers of cybernetics. Back in the 60s and 70s, he wrote some very wonderful books on consciousness and stuff. And he came out with this one page. This is a one-page paper that changed everything. You know, from this, that was the initial condition, you know, the perturbation it created that changed everything. I'm gonna try to tell you a little bit what we're trying to understand. And they asked this question, you know, what is the chance that a large system will be stable? because they were thinking about the brain back in those days. No, still are today. But they say if a large system is assembled, connected at random, or has grown haphazardly, should be expected to be stable or unstable. And how does the expectation change as n, the number of variables, tends to infinity? So that was a question. So they say, well, let's suppose we have a vector of variables. Each one is has its own dynamics, and we can describe this. Eventually, this was they were working with this at the equilibrium. So this is the kind of the what we in ecology call the community matrix, or the Jacobian of the system. And they were trying to solve this and see if it was stable for different connectance. Connectance is the number of connections that the system has. You know, as a percentage, they use it as a percentage of the number of nodes. So they see that there's a sharp decrease in the probability of stability as the number of as the connectance increase, and also the decrease is actually sharper when you have systems which are larger. This is 10 components, this is 7 components, and this is 4 components. So as the number increases, the decrease is sharper, and there's a huge transition in the system. So, and they did this in a VAX machine, you know, running, and they took them forever. So they said, well, we are very limited in terms of computer power. That was back in 1970, and it was a very interesting paper. But then, the stability is like, right, is if you perturb the system within equilibrium, if the system actually collapses or not. That's basically the definition. So if you leave the attractor where it was. So the same question was taken up later on, 2 years later, in the same journal, with a 2-page paper now. They are increasing in complexity. 2 pages. So, and there was Bob May. You may know Bob May because he was one of the, he's a physicist, Australian physicist that started working on ecology back in the 70s and started working with chaos theory and became a big figure. She died a couple of years ago and was one of the most important theoretical ecologists during that time. And May 1972, this paper working with random networks using the same approach as Ashby and Gardner did. And he said, look, what you guys did in terms of computer simulations, actually it can be done more analytically. And he proved that if you do a random network of interactions between entities, and you have alpha, which is the average magnitude of interaction strength among the entities. Suppose this is an ecological system, so this interaction will be how much my biomass affects your biomass. You know, in terms of you find a predator, prey, or a mutualistic interaction. And this is the interaction strength, and S is the number of species, and C again is the connectance of the system, the number of interactions, usually measured as a percent or as a function of the total possible number, which is S squared, in this considering self-interacting system too. So they said it has to be less than 1, okay? And he also said that usually this doesn't happen. You know, so the only way you can have a possibility that a large complex will be stable is if there is a trade-off between interaction strength and connectance, such as you have systems where there you have modules or groups of species that are strongly interacting among themselves, but they loosely interact with the rest. That was kind of the logic. And since this result, people start looking for ways by which ecological systems could be stable and could actually satisfy this condition. And as is obvious from this equation, one of the ways to satisfy this condition is if S is a function of connectance, and it's a decreasing function with a -1, because it would be, if you replace this there, then you have the number of species goes away. and it's no longer dependent on the number of nodes in the system. So stability is no longer dependent on the number of species. And in fact, and if you, this is a recent analysis from Buziello, 2017, okay? And they did an analysis of different networks, and actually the scaling is pretty good. It's 1/s, okay? So in fact, it looked like real networks satisfy these conditions. They also did a dynamical system approach, like a generalized local terror system, and they showed something very interesting. That they say that actually they show that when the system is poised at this stage, the dynamics has multiple attractors, so you have explorability, as they say. So the network can explore different potential solutions. Much more than if they are not in this regime. So that was great, and it was later applied in ecology. There became a big fuss about trying to understand ecological networks. But then things started get a little bit messier because people tried to understand networks, and they started many times by having these nodes, and this node actually is an aggregation of different species. So this network were not very well resolved, and people start showing that if you unlock those nodes and you start including more and more species, all the statistics used to describe the network start changing. So there is a lack of robustness in terms of the patterns, and people start getting a little bit kind of uneasy about networks. And that was understood also because people is saying that the properties of the network are associated with the stability of the system. So we need to have a real good description of these networks, because otherwise we can lose a lot of the interesting dynamics that make this stable. Well, as it turns out, we started using or trying to understand ecosystem by looking at particular interactions and particular food webs, or who eats whom, But this is also a kind of a partial description of the dimension of interaction, because you have mutualistic interactions, you have a lot of non-trophic interactions that Daniel will probably tell you more about, that are increase the complexity of this exponentially, in terms of whatever can be going on. And one of the things that people use to describe these networks, as you may know, is the probability of observing species with K interactions, K linkages, or the degree distribution of the network. And the random expectation is that someone is ringing.
No, that's good.
I thought that someone was coming. Anyhow, so the expectation for a random network is that the degree distribution will be Poisson distribution. And what people start observing is there was a deviation, and this degree distribution were kind of power law, meaning that you have not an exponential decrease, so you have more rare or have fat tail, or the probability of observing highly linked nodes is larger, which means that there are some clustering, as proposed also by the May article in 1972, that things should be clustered, otherwise there will be unstable. But using the same data sets, people say, well, it looks like they are not. I mean, these guys did the community distribution instead of using doing the frequency of the probability distribution. So they did the community distribution and found that actually it was most close to an exponential. So that's when uncertainty starts building up, because there is now uncertainty how you measure the network, or the method you use, you get a different answer, which is not good unless you are working in quantum mechanics. We supposed not to be in that realm. So this lack of robustness soon made apparent that it was composed, that it was very difficult, and also it was composed by other uncertainties in terms of the nodes and the linkages that I will tell you a little bit. The interesting thing, though, is that for other systems that could be more reasonably static or more completely static, like, for example, aerial traffics, know that all the traffic across the airports of the world, you can find that the power law is like excellent fit. There's nothing you can tell that this thing is might be wrong or whatever. You know, the fit is excellent, and the data is very complete. You know, there's no uncertainty or very little in terms of that. And so people also have shown this for the co-authorships networks, you know, where the nodes are authors and the links are co-authorships, so authors that appear in the same paper together, so they are linked. Actually, the first analysis of this was done by a guy with a funny name that I can't recall this day, but it's back in the 60s. He published a paper in Science doing the co-authorship network, and showed that there was a very nice network. And also, these guys now, Newman, pretty much showed that the link distribution also follow a kind of a power. And the same with this sex web, where nodes are people and links are sexual interactions. This was done for 4,700 suites, and so that also follow a power law distribution. So it looked like power laws can be a way of describing this sort of interaction networks at some point, but and also at a more fundamental level for metabolic networks too. This is the metabolic networks, usually reconstructed from the genomes. So you annotate the genomes and you know where this gene is acting, what kind of enzyme it's codifying for, and then you do analysis to actually understand what is the network behind it. And it turns out that the network tends to be also powered. So there might be something to it. I just throw it to you because it might be interesting to pursue at some point. But still there's some important uncertainty that I'll try to explain more. But before that, there is some other way in which networks could be different from or satisfy the condition of vein in terms of being kind of a more cluster. And suppose that we have this very simple bipartite network where you have two classes of nodes. In this case, it's plants and animals who actually disperse their seeds, you know, or this is called, because the interaction is positive, because the bird have a positive interaction with the plants because it's helped to disperse the plant. It's called a mutualistic network, the same as this one, which is a pollination network. So it's our insects or birds that pollinate plants, and that's a positive interaction too. So you can represent this, you know, in different way, but this is plants and all these insects that pollinate those plants. People actually realized that there was a very interesting pattern of the interaction between plants and animals. Suppose you graph, well, this is what you expect if the thing is completely random, okay? But usually if you graph the plants, or if you plot the plants in this matrix, starting with the one that has the largest number of pollinators down, and you do the same with the animal. We started with the animal who actually has the larger number of plants that pollinate down. You find that it satisfies this sort of nested pattern where this plant here, which is highly specific to this pollinator, you know, the pollinator actually is a generalist. And this plant, which is also interact with this this very specialized animal, is also a generalist. So it looks like generalists interact with specialists. So this is an asymmetry that is manifested in this, what they call nested pattern, because this species is netted into this other, into this other, into this other. You see? So this nested pattern of interaction was supposed to actually bring a stability in the system, because a generalist species interact with a specialist. If the specialist, suppose the specialist disappears, then nothing happens because this other species has a lot of other interactors in the system. You know, so that's built a sort of resilience into the network. And it was also shown using dynamical system and approach and local terra equations that this relationship actually fosters stability. And this is the real network. By the way, they have been done for many other different kind of networks. For example, if you put businesses in different counties, different counties in Paris or different counties in France or Chile, and you put here banks, hairdressers, washing places, theater, blah, 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 blah. It's also nested. And sometimes you find that there is a casino here in a place that has 2,000 inhabitants, so that business is doomed, it's gonna fail. So the same here, you have these unexpected presences that you can attach high probability of extinction. But anyhow, so there are other stochastic or other patterns that can be manifested in this network that satisfy these conditions too. So let me tell you a little bit about the indeterminacy of these ecological interactions in networks. And this is a paper done by this ecologist, theoretical ecologist, Peter Jatsis, one of the influential figures. And he came out with this idea a long while ago, back in the 1980s, which I think is very potent, very, very nice. Because he works with this, dynamical system for different species indexed by I, and he shows that the equilibrium is going to be determined by a matrix of this form, which is the community matrix, measure at equilibrium. The same one that Gardner used and May used is all this kind of linear stability. And he then asked the question, well, what happens if I start adding something to a particular species? So I start adding individuals to that particular node. What's going to happen with the rest of the network? And he came out with this solution that say, well, whatever happens, the direct interactions under some conditions are going to be measured by these, the inverse of the interactive matrix. So you take the inverse, and the inverse will give you, he showed that, will give you which are going to be the interactions on a particular node of doing this experiment on some other node. Okay, and he shows that there are fundamental kind of uncertainties in what this propagation will be, because if you take different good realization of the system, you get a picture like this, or you can get a picture like this. So he says, well, there is a fundamental indeterminacy in terms of the impact that a particular node will have on other nodes in the networks, and also in terms of the particular network, the particular impact or interaction strength, how important are they going to be in affecting the dynamic of the network. So you have particular, uncertainty in the interaction strengths, and also you have uncertainty in how they propagate through the network. Okay, so that was kind of the state of the art, and then it came this realization, in part because of this, that if you want to reconstruct a network, which is especially important when you are working, for example, with the bacterial network in the soil or in the sea, where you don't have all the species, you have genomes, and there might be many rare species that might be important. you have to deal somehow with this fundamental stochasticity in these networks. And so that's a real kind of a, I would say, hot topic these days. There was just a paper, I will show it to you a little bit, trying to apply Bayesian statistics to understand this network structure. And what we have done is just go into the stochastic approach, and just to extreme the case, just by doing stochastic differential equation like this one here we did with Rolando Rebolledo, and which actually allow us to see how stochasticity can impact in this network, start trying to build a theory, but it's very difficult to get to any analytical result for example, invariant distribution or something like that. This is a very particular kind of network where you have an interaction term here between the biomass of species X and Y, and this is the other terms. This is the decrease in biomass due to the interaction with other species. And here we have a Brownian with a coefficient sigma, but also Rebolledo wanted to do something different, so he added another noise term, so he added a Poisson process here that actually is like jumps. You have the Brownian, and eventually you have a jump, in the dynamics. And this jump is here to actually simulate particular perturbations that might come into the system, like a particular weather event, a flood, or something, a fire that might impact on the dynamics. And he could prove that there was, existence and unicity of the distribution, but of the solution, but nothing else. And this is part of the dynamics that you can see for a particular realization of this network. So it's very complicated. If this is a good description to actually come from here to infer the topology, because you're going to have different kind of trajectories there. And things get more complicated when you put more than one interaction. We were working with predator-prey, and also including some non-trophic interactions, like in this web that Daniel will tell you about more, where you have not only the trophic links, but also the interaction between links and non-trophic interaction like competition, mutualism. So, and this is one of the latest approaches to reconstruct networks done for plant pollinator network using Bayesian approaches, which is very good because this one actually used information about all the components of the network to infer a particular component dynamics. Not only some of the others that were available used pairwise or pieces of the network to infer something. This one used the whole network to make inferences on the dynamics of the network. Well, let me switch gears a little bit here and tell you a little bit about something we have been doing, which is trying to work in ecology to do some interaction or integration of different theories in ecology. And one of them is network theory. So we want to build models that are compatible among themselves, because we think that, since we are studying the same big picture systems, it cannot be that your theory implies something completely nonsensical to this other dynamic of the ecosystem. It cannot be that the theory for abundance is nonsensical to people working with network theory or people working with species richness theories. There has to be some integration. They have to be meaningful, and if they touch, they have to be logical or agreeable touch. So we have been working on this for a long time. We have a network of people working with us, and we have been trying to advance theory using mathematics as one of the tools to do that, because we think that mathematics is fundamental to actually do this job because of lack of ambiguity it conveys. So have you seen this before? You should bow. No, this was one of the most amazing relationships we had in ecology. It says that the number of species increases with the area that you sample raised to an exponent k. And actually, it started by people doing quadrats. You know, you measure how many species here, then you increase the quadrant, and you start increasing the quadrant, and the thing goes as a power law. And people start looking then at islands. You go to a small island, you count the number of ants. Then you go to a bigger one, count the ants, and a bigger one. And there is this power law, which is very interesting, that there are no agreed kind of explanation for it. You have, this is the very simple way of representing it. It might have different phases, but it's a fundamental pattern that inspired these guys. MacArthur was a mathematician. Actually, MacArthur was one of the fathers of ecology. He was a mathematician. He wanted to put ecology into a quantitative food back in the 70s. He developed a lot of models, and this is one of the models he developed with Wilson that gave rise to this book, which is a monography of population biology. It's the #1 monography. This series is one of the best series for ecology books ever, and it was the first book back in 73. So he never developed an explanation for the other equation, but he developed a theory for how the number of species in an island change, and use this master equation approach. Very simple. You have a birth-death process. You have a probability of a colonization, and you have a probability of extinction. So the way this probability changes is because nothing happens, or because you have S-1 species and one colonized the island, or because you have S+1 and one died, or one extinct. Okay, very simple. Actually, they never solved for the environment of this. For some reason, they were not preoccupied with that. Later on, people start working on it. And here you have rates. This is colonization. This is extinction. And you see, it predicted this particular distribution. That was fine. But later on, back in 2000, someone else came out, and it was Steve Hubble, and applied the same equation, but now at the level of individuals, saying, what is the probability of observing a particular species with n individuals? I said, well, depends on the probability. Maybe you have n+1 and then you have a death, or you have n-1 and you have a birth, or nothing happened because you already had k species, n individual. So then they assumed that all the species were equal on the per capita basis, and there was density independence, and they compute the invariant distribution, and was the Fisher distribution of abundance. And this was a prediction from this theory that assumes that all species are equal, or all individuals are equal in terms of per capita birth and death rates. Okay, so that's what is neutral. It's kind of the same as the neutral theory in genetics, where genes are assumed to be having equal impact on fitness. So he derived this. The problem is that you cannot have one master equation for the number of species and another for the number of individuals. Because species are made out of individuals. So unless every species has one individual, these two things cannot be true at the same time, because one will be non-Markovian, and you are assuming Markovianity. Okay, so this is one thing that people have been doing models and here and there, trying to explain stuff, and it cannot be. You know, that's my favorite example of why we need mathematical logic in here. Otherwise, we will keep wasting our time trying to work with these two as independent, and they are not. There are ways to actually solve this problem. You can say, well, let's make the number of species to be unobservable on this Markov process, and then you can derive a distribution. But there is some other ways, and the other way is forget about numbers and go into the continuous limit of proportion and do a diffusion approximation to this, something that has a long history in biology with Seward Wright, Fisher, and of course, Kolmogorov, who actually told these guys, you know, Fisher and Seward Wright, and particularly Fisher, that his results that were published in Russian, and eventually Kolmogorov published a short paper in PNAS showing this, what Fisher called the differential equation, the partial differential equation for selection, showing the theory strongly. And what they derived, and pretty much this is the derivation that did Seward Wright. Seward Wright always thought of a structure of populations. So you have subpopulations interacting, exchanging individuals. So unlike Fisher, who liked to work with a single large population, he liked to work with this structured population. He showed that the invariant distribution for the gene frequencies, one-dimensional, was this one, a beta distribution, you know, under certain assumption, and which is kind of the model people pretty much use today. So we did the same. We actually moved from numbers to frequencies to this, and that will solve the incompatibility between the richness and the abundance distribution. We did it with the help of Rolando, who actually, for his dissertation, he invented, or he derived several theorems to actually solve these problems of moving from the discrete to the continuous. And basically, just to make the story short, we have a model like this. So this is in dashed line because it's our focal system. Suppose we are looking at a system that is our focal system, but we know that it's not defined completely and interacts with environments. And we are going to say that that interaction with the environment or everything else about the system that we don't know is going to be in the noises. And we transform these numbers into proportions. And we know that when this numbers is huge, eventually will converge to this stochastic differential equation, and that this eventually equation at any point will be compatible with this Fokker-Pan equation, which in the time limit will eventually converge to an invariant rho infinity here, which in this case, for this kind of coefficients, very simple one, very than process, additive, and here the CJs represent everything else that we don't know about the system, which is included in the noises. That's where the C is in the coefficient of the noise actually converge to a beta distribution, too. So that's what I was kind of so keen on, the Dirichlet and the beta distributions. And when you apply it to understand the abundant distribution of moth, tropical trees, coral reefs, and different kind of systems, actually the fit is pretty good, as good as it could be using some other models. So, and we also came out with the coefficients depends on the type of systems. One of them, particularly alpha, is more associated with colonization, and beta is more associated with speciation of the number of species that you find in the system. So just to wrap up, there is some fundamental stochasticity and uncertainty in networks that can be tried to rescue by moving the theory of networks into more stochastic theory. There are statistical methods that allow you to reconstruct the network structure, but also there are ways of trying to move into a stochastic description that we hope to apply this theory now to the in the context of networks, and to build a theory that might be compatible with this one in terms of predicting distribution like this, but now based on the interaction of ecological networks. Thank you. Almost there.
Thank you. Are there questions or comments?
Questions, comments? Tomatoes? Rice?
It is Pablo Marquez who asks questions. So,
I don't even have a question for myself. So
no,
that's terrible.
By the way, I found the
I
door.
know. No,
There was a door.
that was a door. Yeah.
Which was this door?
The blue door.
Yes, it was not the door of the torture places in Chile, because I would.
No, I don't want to
once.
open that door.
Oh, yeah. Okay.
No, that one is closed.
Okay.
Yeah. No, that was a door in Santa Fe.
Okay.
It was a nice, I like it.
There are those torture places which are still with the plague in
Yeah.
Chile.
Yeah.
Okay.
But that's a different stochastic process.
Yes.
That's pure death.
Yes.
Yeah.
So I don't know. We may just thank you and
go to the different
and
the next.
okay and go to. I just make something here.
Don't leave anything here. No.
I just cut a bit the record.
Okay.
Okay. So here. Is that okay now?
It's perfect.
Are we recording? I think we did.
Let me wash it.
I think
Yes, I think
It's
it's
recording.
recording.
Okay.
Yes.
So Daniel, thank you for coming to us, and I'm happy to welcome you.
No, I want to thank you for the invitation. And hello, everyone. Just let me know if I'm speaking louder enough. I have some short troubles, but I will do my best. Thank you for Paulo for the invitation to be here, and also thank you, Paulo, for that invitation. I want to present a little part of my PhD thesis, which is mainly in the interplay between what Paulo called before the interactions, and the idea that species interact in several ways. So, as we know, I don't know if you will see those pictures before, but those pictures correctly. But as all we know, the species in nature interact and coexist at the same time. And those interactions affect each other's population. And that happened in terrestrial ecosystems, but also in marine ecosystems, in every ecosystem in nature, okay? And those interactions could be trophic, in which we have a predator who eats or who feeds on a prey, affecting the negatively abundance, affecting the population of the prey negatively, but also we have no-trophic interactions. And the idea of these no-trophic interactions is that we not based on that idea of whom is whom, okay? And those non-trophic interactions could be positive, as one example of that is here. And a bee which is pollinating a flower, and it's improving the capability of dispersal of the seed of the flower or of the pollen of the flower. And also, we have another example here in which we have an anemone, which provide shelter for a clownfish, and that shelter provide reduce the mortality due to predation on the clownfish. And as well as we have positive non-trophic interactions, we have negative ones, sorry. And in rocky shore ecosystems, we have that some barnacles, mussels compete for a space. So the space there in that system are limited resources. And if you see another example here, there are several plants in a particular place that compete for the sunlight, but also for the soil nutrients and so on. So when we represent those interactions, the patterns just start to emerge. And I want to show you this, that is the same image that Pablo showed before, because in this paper, Sonia Kefi highlight the idea that trophic and non-trophic interactions act between pairs of species simultaneously. And also, they, in this same paper, trying to incorporate those non-trophic interactions into network theory. Just to, after this, I want to make a parenthesis here. After this, mainly the food networks was based mainly on the idea of whom eat whom, just to be clear. And with this, they attempt to include the non-trophic interactions on that network theory. Okay, so they proposed that nodes, particular nodes in networks, could be affected by modifying directly the feeding of other species, for example, and in which we have an example again. We as I told you before, this anemone provides a refuge for clownfish, and in that way, the clownfish, the predation pressure of the clownfish is reduced. But also, they show that another way to include non-trophic interactions in theory is to modify non-feeding attributes. And again, I'm back to the example of the bee and the queen stage. Okay. And later, Sonia Kefi and colleagues, using the Chilean intertidal network, found that the links among between those between the interacting pairs of the species could be constrained. So we have few pairs of species that are involved in more than 2 types of interactions simultaneously. And one of the particular cases in which this happens, in which there are more than 2 types of interactions simultaneously, is in this species, which is beyond a mussel in the intertidal system. I'm going to show you some picture before, after this. And also crab, which is Acanthocytus asledi. And those species, okay, this is a mussel, and inside the mussel, there is a crab. Just bear with me, please. But those species are involved in several types of interactions simultaneously. And I'm going back to this picture later to explain a little bit more so what's going on inside this system. But the fate or the abundance of the population in particular places do not rely just on on how the species interact. They also relies on physical processes. In terrestrial system, for example, the wind could drive and modulate the dispersal of the seeds of plants along a gradient of the space. And in marine systems, we have something similar or pretty similar, which are the marine currents. And those marine currents transport kelp seeds or transport marine invertebrates or fishes larvae along the gradient of the coast, for example. So I want to be brief here because I want to just to point out that part of the inspiration of my work is based on how those kind of systems or how much those complex systems are sensitive to environmental variation, such as temperature or such as recruitment. And to face some of my questions, I choose a simple pairwise, and I will quotate a simple pairwise biological system in which we have, again, a crab which preys on a small mussel, okay, which feeds on a small mussel. So they are involved in a predation relationship. And the thing is that this mussel is a really dominant organism in the Chilean rocky shore ecosystem. And the aggregation of that mussel generates three-dimensional matrices along the coast. And those matrices are used by several kinds of organisms to protect themselves from predators. And one of the species that use that as that use that matrices is also it's one of its main predators. So they are involved in more than one type of interaction. And I'm going to call that interaction refugee provision. Yeah, I'm going to explain a little bit more later. But also, both species have complex life cycles in which they free larvae to the water column. Then the larvae are dispersed by the currents, and that larvae from the particular sites are dispersed together with larvae from other places along the gradient, along the spatial gradient. And after they find a place to arrive to the rocky shore, they arrive, they settle. So, or they recruit. I'm going to explain this term here, recruit. And I'm going to say that the recruitment is or represent the idea of a species from the column, or a larvae species from the water column arrives to a particular place in the rocky shore. Okay. The thing here is that we have evidence that we know that this predator do not recruit on bare rock on the intertidal ecosystem, and instead needs the prey or the matrices of the prey to recruit. So they are also involved in a third kind of interaction, which is the recruitment facilitation. So to summarize, these pairwise system species are involved in recruitment facilitation, and I wanted to show you better these metrics of Parameciums corporatus, but anyway, and this interaction could be understood as an interaction in which the prey affect an attribute, a non-fitting attribute on the predator, but also they are involved in the second non-trophic interaction, which is the refuge provision and protecting the prey from the possible predators. And in this case, this kind of interaction is different from the other because it's affecting a feeding attribute on the possible on a possible network. Okay, so we have some evidence, some experimental evidence to know a little bit what is happening here. And Estela Januario, in her PhD thesis, found that, and on the x-axis, I'm going to plot the settlement or recruitment rate of the predator. And she found that there is a preference for the predator to recruit on mussel beds, are those with gray bars, instead of recruit on bare rock. And this pattern could be affected by the mussel cover along the particular place, the intertidal zone. And also, and here we can conclude that Acanthocyclus, the predator, needs a facilitator in order to recruit. But also, Sergio Navarrete, Myrtala Parraguean inhibitors show that the crab density, and I'm talking about the crab in its adult stage, has not a the density of the crab is not a linear function of the mussel cover along the rocky shore ecosystem. So this highlight the idea that in order to this crab to exist in high densities, needs to reach, needs that the mussel cover reach a particular density to start being successful in a particular place. So with Sergio Navarrete, Alexander Genoa, and Sergio Rojas, we use modeling, dynamical modeling, to ask whether non-trophic interactions or these non-trophic interactions have similar effects on the predator and on the prey. But also we ask whether those MPIs or those non-trophic interactions will foster the coexistence of both species in places where the recruitment is limited. And we start in a simple point, and our simple starting point was a model in which we have on top the prey dynamics and on bottom the predator dynamics. A model in which the growth part is modeled as an open system. And this means that unlike the common, for example, Lotka-Volterra or predator prey models before, the dynamic or the incoming of the individuals to the system do not depend on the local dynamics. Instead, depends on these parameters, which are the recruitment rate for the prey and the recruitment rate for the predator. And also, we are modeling here the space as a limiting resource. Okay, we have then natural mortality for both species. And finally, to include the predation effect, we have this type 2 functional response that represents the idea that the consumption of the predator relies on the prey density. And we estimate the parameters for this model using experimental control in a sim. And then I'm going to show you how the prey density and the predator density, the prey up top and the predator density could variate along the recruitment rate of the predator along the y-axis, and on the x-axis, the prey recruitment rate. And what we see here is that in this particular place, when the predator recruitment is high and the prey recruitment is pretty low, we have almost an extinction of the prey. But as well as we start to decrease the predator recruitment and increase the prey recruitment rate, the prey density starts to increase. Okay. However, here in regarding to the predator, we have a variation just that relies on how much of the incoming of the larvae arrives to the place. Okay, and then we want to include the non-trophic interactions, and to include them, we use the approach using before for Kefi, for Kefi. And in that approach, we think, or we modify the recruitment rate of the predator as a function that increase when the prey density increase. So in this particular case, the predator recruitment rate will range between Cmax and Cmin. And the thing here is that the prey need to have a particular density in order to for the predator recruitment start to increase. And also for include the refugee provision, we modify the mortality rate of the predator, and we use a similar approach, but in this case we have a decreasing sigmoidal function in which we have a maximum mortality when we do not have prey in the system, but as well as the prey density reach a threshold, the mortality start to decrease until a minimum mortality rate. Okay, so to evaluate where the non-trophic interaction foster the coexistence in limited recruitment environment, we simulate a scenario in which we have low prey recruitment rate, low predator recruitment rate, and high predator mortality rate. This is kind of a pretty bad community for everyone. And we plot the strength of the interactions. And this strength of the interactions are just the magnitude among Cmax minus the Cmin, and the strength of the recruitment facilitation, and the relation between Mmax and Mmin on the strength of the refugee provision. And here I want you to know that on this origin, we will have the model just with the trophic interaction, and we could see how in these places where we just have the trophic interaction, the prey reaches high or the highest density, but the predator is almost extinct. But once we start to increase the intensity of both interactions, that density start to change and start to increase. And start to increase for both the prey and the predator. But here, the thing is that we do not have idea or a fundamental basis to know which is the strength of the interaction in the system. But we could see that we could reach a point in both species where by action of the strength of those interactions, the prey will be almost extinct. And to understand the mechanisms of the non-trophic interactions, we just estimate a ratio which is the subtraction of the density without the non-trophic interaction to the density with non-trophic interaction, and divided by the density of the non-trophic interaction, and for both the predator and the prey. And we with this, we want to observe if the values are positive and increasing the density due to the effect of the non-trophic interactions compared with the model without the non-trophic interaction. If the value is negative, we will observe a decrease on the density by action of that non-trophic interactions. And we plotted again the ratio density on the y-axis for the predator, for the prey, and for the predator along the x-axis of the strength of recruitment facilitation in this particular case. And we saw that when we have the maximum recruitment facilitation, the predation ratio could increase up to the 50%, but the prey density decreased 50% again. And I want to explore these results further because I think that we will have here a plateau, a rich plateau, and that plateau could be due to the dependency of the predator to the existence of the prey in the system to arrive to a particular place that we are modeling. And here we have the same approach, but for the refugee provision. And we could see that a pattern is quite similar, but the magnitude change. And in the case of the prey, we could have a high decrease, almost at the 35% of the density compared with the density with the non-trophic interactions, and sorry, without the non-trophic interaction, and that the same increase in the predator density. So here the thing is that I think that in this particular interaction we couldn't have a plateau. Instead, we could have, just by action of the refugee provision, we could have an extinction of the system. And that's pretty much difference what I think to happen here. And finally, I want to show you how is the performance of the model in order to assess how the spaces of coexistence for both species will be moved due to the non-trophic interactions. And again, I am back to this plot in which we have the effect of the trophic interaction. And here I have the effect of both non-trophic interactions in the prey density and in the predator density. And what I want you to note is that in those particular places or spaces of coexistence, when we have low predator larval availability. The non-trophic interactions foster the increase of the predator density, but in some of those spaces here, we could have a sharp decrease in the prey density, but we could have also a coexistence in medium to high densities on those of the space of the parameters. So in summary, we found that we have different effects of both kind of interactions on the predator and on the prey population. And we found that the NTIs foster the coexistence when the recruitment is limited. And finally, I want to just to give you this idea and just to show you how the colors change, increasing the densities when I include or increase or compare the models with non-trophic interactions, just with the recruitment facilitation, just with refugee provision, and with both, and just note the colors, how the prey density and the predator density will change along this axis of variations. And what's next? We were talking about what we want to do once we finish with this part. And what we are thinking are to assess the sensitivity of the system to the temperature. And we started experimentally with some of that. And we assessing the effect of the temperature on the functional response. Also, we think that we could frame this work on a metapopulation framework, and also due to the connectance of the species that we are working with, we can assess the community scales consequence in a bigger network. Okay. And finally, I want to thank all the people which helped me in some of these experiments, but also give me their advices. Thank you. Yes, Las Cruces.
Other questions? For sure. So, Daniel, you are modeling. This is a question that nobody will understand. That I will try to explain it. So you are basically modeling the system inside Las Cruces.
It's a particular place. Yes, it is. But we wanted to start at that place, at a particular place, and thinking on the recruitment as a gradient of the variance on the space. But later we wanted to include more variation along that axis of space.
And for the people, I mean, Las Cruces is a reserve. So inside Las Cruces, there is no human impact. And it's kind of a different system. I mean, particularly the cover of muscles is reduced, I guess, as compared to outside. So inside, you have a very nice experimental setup in terms of having a kind of a perturbation in the system that you could use the model to assess the impact of humans.
Yes.
Chapter 4.
I will think.
Are there any questions? I have another one. Yes, I mean, eventually you can start from any node in that network and do the same thing as you are doing, like looking around and see which is the closest node I interact with and start adding other nodes, which could actually be very interesting to assess which are the more important nodes affecting the dynamics of a particular node. So have you considered or thought about eventually using this approach to just go beyond? You have the mussel, you have the crab, you might include the other species of crab, you can include the predator of the crab or the predator of the mussels, or the predator like the local or some other species. And I think people usually do that in the context of it's called this weird name is the Markov blankets, to see which are the different number of nodes in your neighborhood that affect the dynamics of a particular node. This could be kind of interesting.
Yes, I'll be thinking of that because that's one of the ideas that I have, but I first need to finish the dissertation of this part of my work. But perhaps in a postdoc, also I could be inspired on a paper that I've been reading before, which is of Ian Donahue and Sonia Kefi and Alexander Benoit, which they do something like that, and they assess how much the model fits the reality. So they have also experiments in which they control all the nodes in a small network and a network of like 5 species. So I think that that's something like that you are suggesting. But yes, the answer is yes. I'm thinking of that. Thanks to you.
No more questions, Fabrice?
I have a question from Zoom.
So, please.
Okay.
Yeah, this was a great talk. It was really interesting. Here, let me turn on my video. I was wondering, yeah, so it's interesting the way that you compared or included non-trophic interactions between the predator and prey. And I was wondering if there's some analogy that you might be able to draw between these non-trophic interactions and say like optimal foraging theory, like how especially with thinking about this differential recruitment of predators in places where their prey are.
But you mean something related with the size of the prey?
No, thinking about how I think if we think about a more mobile predator that can travel longer distances, we think about maybe that predator will go to places where their prey are high concentration and eat there. In this case, the predators, they're mobile, but they're limited, right? So they're going to live their whole life in an area, in whatever area they originally land, is one of your assumptions.
Okay. No, I didn't think in that, but it's pretty interesting. I think I could think a little bit more on that.
Okay.
Sorry for not.
No. Yeah. Thanks for the interesting and inspiring talk.
Thanks to you.
So I think we may just thank you again for the presentation. And I stop recording 1 moment, waiting for the next talk. Wait. That's really a pity that you are not with us, but COVID decided something else. So I am a bit sad that you are not with. You were not able to come and visit us in Paris. But anyway, I thank you for attending the conference and giving your talk. So thank you, Mara, for coming.
Yeah, thank you all for the invitation. I'm also very sad to not be there in person. But I hope that I'll be able to participate still. And I know it can be hard with a virtual presenter, but Paul, if there's any questions that come up during my talk, feel free to interrupt, or anybody. It might be a little harder for me to hear anybody else who's farther away from the computer. Yeah, so my talk overall is on this topic of network inference, coming mostly from the perspective of an ecologist here, and thinking about how can we infer ecological networks using the existing data, spatiotemporal data. I'll be using observations and also a stochastic model, and it builds on what you've just heard from Pablo and Danielle. This is work so I'm currently a postdoc at Scripps Institution of Oceanography, University of California, San Diego. This is work that I did or started as a Fulbright fellow, where I was fortunate enough to be able to work with Pablo, Rolando Rebolledo, and Sergio Navarrete, and many others at La Católica in Chile. And so just a summary, and this relates to some of what you heard from Pablo, but this question of ecological networks is really fundamental to the study of ecology. And so the question the main question I see is asking, what is the relationship between species co-occurrence in space and time? So something that we can observe easily, and interactions between species. With the interactions between species, what we want to put into our models, and those models then give us the capacity to predict how ecosystems might change. And so there are a number embedded in this big question. There are a number of ecological and statistical challenges, and I see them often in some sense going together. So on the ecological side, we have this challenge. How does the structure of, is everything okay? Can you hear me?
No, I was just wondering if everyone can hear, and I was thinking about what we can do.
Okay, can everybody hear or no?
I'm sorry. That's okay? Okay. Maybe I should just turn the computer, maybe it could be a bit better.
Sure. Yeah, I see the ceiling now, but can you all hear me better? Okay, great. Yeah, great to see you all. Let's see. Okay, yeah, so saying we have these challenges, these ecological challenges and these statistical challenges, and find that they are related to each other. So where our uncertainty about ecology is related to our to the mathematical statistical tools that we have available to us. As Pablo was saying, this idea that theory is really essential to our ability to advance ecology. So on the one side, we have this question, how does the structure of ecological interaction networks affect ecosystem functioning and stability? You heard about that from Pablo. And then the statistical challenge that's related, which is one that I'll be addressing in this talk, is how does the network architecture, so how does the nature of these interactions affect our ability to actually reconstruct those ecological networks? So to know what those interactions are from the available observations of species distributions. The other, what is the role on the ecological side? So what is the role of these non-trophic interactions, weak interactions, rare species in assembly of community and these distributions of species? On the statistical side, it's a little bit more loosely related, but there's a really fundamental challenge that the time series analysis tools and our understanding of random matrix theory, when we have non-Gaussian distributions, is relatively limited. And that relates especially to this idea that species abundance distributions are not Gaussian, and distributions of interaction strengths are also not Gaussian, typically in ecological systems. And then a final challenge, although I think we could go on, and I'm sure we will during this workshop, of the many ecological and statistical challenges, but one that's really core here is how do organisms interact both with their biotic environments, with the other organisms there, but also their abiotic environment, so the ambient environmental conditions. And I think this is revisiting these topics of ecological networks becomes increasingly relevant as we have more tools for big data analysis, but also as ecology develops more tools and becomes a big data field. And my interest in ecological networks comes partly, I think I consider myself broadly a scientist, but my training or my PhD is in oceanography. And I think about especially marine microbial systems. So in the ocean where microbes are performing a lot of the primary production, a lot of the ecosystem is microscopic. And that's challenging because it's really difficult to observe interactions between microscopic organisms, and the number of organisms and distinct species that we have is just enormous compared with terrestrial systems. And so there's this real imperative to have these statistical tools, such as ecological networks, to use. And I think to some extent they have been successful as hypothesis generation tools. But there's still this challenge of what do these networks mean? What are the ways that we can best reconstruct networks so that they give us the species interactions? And so I did this work in Chile, which was, I think it's a really unique and special site to study ecological networks for a number of reasons. One is this fantastic data set. So I have to thank Sergio, Evie, Bernardo, and the large number of people who have put effort into collecting these time series of data. So I used data from 1998 through 2013, covering the central Chilean coast. The other, you've heard about this known ecological network. Sonia Kepe was the lead author on this publication, where between the 110 species on the coast, the trophic interactions are known, the positive non-trophic and negative non-trophic interactions are known. And so there's this really complete knowledge of the system that makes it really special. And then the other is intertidal system generally, I think, is a good place to test these series of species assembly. And there's been a lot of ecological theory that has come out of intertidal systems. And part of that is because there's this environmental gradient. So at the low tide, so at the low tide, that's an area that's covered by water most of the time. Whereas at the high tide, most of the time that's out of the water. And then the mid tide is kind of in between. And so we tend to think of a higher environmental stress in this high tide region because there's more exposure potentially to predators like birds. There is, and there's also increased risk of drying out that the organisms need to deal with throughout the day. And so what I did is I took all of this data that we had available from these intertidal samples, and I applied pairwise correlation techniques to say, a lot of the ecological network reconstruction is based on pairwise correlation or covariance. If we apply those techniques to this system where we have these known interactions, are we reproducing the interactions from this network that I began to call an association network? So the network that comes from our statistical inference. And so here's the mapping of the association network onto the known interactions. And so here, each of these nodes in the network is showing the species and its preferred tidal height. They have habitats, even though it's a very small spatial area. And the links is showing when there's a known link, but the color and the size is, so it's a known link that also shows up in the association network. The blue links here are positive associations, so they're preferentially found together. And the red links are negative, so preferentially not found together. And this is for the positive non-trophic interaction network. So these are species that are known to have a positive non-trophic interaction. Are they found together? And so I think we see some organization related to which species just have more positive non-trophic interactions, and these are these habitat forming species. So you heard about this mussel, P. purpuratus, from Danielle. There are also some algae that play a role in providing refuge and habitat. So this Lawsonia and the Mazarelas. That you see as being particularly highly connected and having positive associations with the species to which they're connected. So I want to provide just one example because I think this looks great in some ways. You say, yeah, we actually are able to detect these positive non-trophic interactions. So let's look at this Lawsonia kelp, which provides habitat for the limpets, Scurium. And then, and we see that those two have a really strong, so large effect size, bold arrow, positive non-trophic interaction. There's this red algae, and those both actually prefer the low intertidal, so the area that's under the water most of the day. But we find that there's this red algae that prefers the mid-tide area, and although it's supposed to have a positive non-trophic interaction with the limpet Scuria, we find that there's actually a negative non-trophic interaction. I think there's two reasons why that could be. One is they actually don't have exactly the same tidal preference, and so although there's a known positive non-trophic interaction, maybe it's a less frequent interaction. But the other is that this red algae, the Mazella, actually has a competitive relationship with the kelp, Lawsonia. So they're often not found together. And so this could be an indirect interaction being that shows up here as a direct interaction mediated by Scuria, when in fact it could just be an outcome of this negative non-trophic interaction between these two algae species. I also, since we just heard from Daniela about the impact of this mussel, we see that this mussel, P. purpuratus, has a very strong positive associations with a lot of species in this network. I highlighted the two crab species that Danielle's talk focused on, A. hassleri and A. gay, and I think it's ends up complicating the story a little bit, potentially because of this existing trophic interaction, where all these interactions have very small effect size, and for one of the species, A. gaiae, it's positive, and for A. hassleri, it's actually slightly negative association, which I think Danielle's modeling work can help us understand. But that's based on, do we know these interactions? And the challenge that I'm working on is one of the challenges that Danielle said, which is, although we know that these positive non-trophic interactions exist, we don't necessarily know the strengths of those interactions, and so it's difficult to know which of the parameter range he was using is the correct one. And I think what we're seeing here is there's actually a very limited ability to use this co-occurrence data to actually infer that. Yeah, and so this is the positive non-trophic interaction network, but we constructed the networks using from the trophic to compare, and so we compare this reconstructed network now to the trophic interactions as well. And so when we do that, we find that they're actually not capturing very many associations. But one of the things I realized was that the species often share a habitat, so the low tide, mid tide, or high tide region with their predator, or with their predators or their prey. And so when I use all the samples, we say we capture about 40% of the trophic interactions. Whether we knew that they were trophic interactions, that's another question. So this is the sensitivity, the detection sensitivity. But then I separated out the data. I did these association networks only using the high tide samples, only using the mid tide samples, and only using the low tide samples to avoid this complicating impact of a shared habitat. And we find that our sensitivity is dramatically reduced when we do that. And this doesn't just have to do with the sample size. I did those statistical tests. And the result of this is we find that the environment is really having a strong mediating influence. And that these environmental associations, or these niche associations, are actually what we're able to detect. Now, if we found similar results for the negative non-trophic interactions, but the positive non-trophic interactions, and this is the reason why I use this as an example, we find it's actually slightly different. With the positive non-trophic interactions, we actually find that there's actually a fairly high ability, or that these are relatively well detected. So when I use all of the samples, we're detecting 77% of them using this association network. And when I separate out the samples, by tidal level, we see that that power is reduced, but the sensitivity is still much higher than either the negative non-trophic or the trophic interactions. And I think this is a really interesting and compelling result. And part of that is because I think that it's consistent with this idea that the environment, so the species niche, is what is most detectable using the co-occurrence of species in space and time. But positive non-trophic interactions are biotic interactions that affect species niches. So the habitat for a species is not just rock at low intertidal, it is rock covered by kelp. And that's what this is detecting by association. Yeah, so my motivation for doing this was to understand these networks between microorganisms. So we've been looking at these macroscopic organisms where we actually know their interactions. And so these network techniques are not necessarily providing additional information about the ecology, although in some sense, we actually were able to look at how these interactions have an influence on community assembly. But I was interested in, can we apply some of these findings from the macroscopic organisms to the microbial networks? And Clara Arboleda Baena, who's currently a postdoc, led a paper where we compare the macroscopic organisms in Las Cruces and the interaction networks, or the association networks between those, to association networks between biofilm communities, so microbes in the same intertidal system at Las Cruces. And I think what's remarkable is we actually find pretty similar patterns in these networks overall, where we find strong negative association between the low intertidal and the mid intertidal and high intertidal. So there's exclusion of species based on their environmental niche. But there are some differences. So one difference is that we actually have these highly connected networks with strongly positive association between species that prefer the high intertidal zone. Whereas for the microscopic organisms, we have strong positive associations between species that prefer the low intertidal zone. And so the high intertidal zone for microscopic organisms is an area of high stress. But this inference led me to think maybe the low intertidal zone is actually one where biotic interactions matter a lot because it's a high stress environment for microbes. And so it helps with some hypothesis generation once we have some sense of what do these networks mean. And so the summary of this observational part is saying we found that co-occurrence networks do not reproduce interaction networks, but they still do seem to provide interesting and interpretable information about community assembly, even if it's not directly telling us how species are interacting with each other. We find that environmental variability, so the environment may be leaving a more discernible signal in spatial co-occurrence patterns than the biotic interactions alone. And we find that these habitat engineering species, so these species that are especially important in these positive non-trophic interactions, may leave a more detectable signal than other interaction types because they expand or create niches for these species which they interact. And so this study was using pairwise interactions, and I should say it was on the slides, but this was published in Ecology in 2018. But it led us to this question: are whole communities more detectable than pairwise interactions alone? And this relates to the statistical challenge of how the ecological network architecture affects the ability to reconstruct ecological networks. And so for this, we're going to turn to the stochastic model you heard a little bit about from Pablo, that was published in these 2017 and 2019 papers, and this work was published in 2020 in Proceedings of the Royal Society A. But essentially what we're doing here is we're taking a Lacca-Volterra model, a Lacca-Volterra predator-prey model, and we're adding a diffusion term that's a stochastic birth-death process. And for this, I'm going to take small modules at first, small network modules that have a range of architectures, and these become this interaction matrix D. So in this one, we have a 3-chain trophic level and so all of these have 4 species and are connected. And so the workflow, which mirrors the workflow that one might have using observational data, is we have this true network, we simulate abundance. Now this is time series alone. And from that, we then use a couple of different network reconstruction techniques. So covariance, which is pairwise, take the inverse covariance matrix, which is a maximum entropy technique that detects the whole network at once. And from that, we get these species matrices that then give us a reconstructed network based on the significant interactions. And this reconstructed network, in this particular case, we see is not the same as the true network, and we'll find that that's actually a fairly general, if unfortunate, result. And this relates to this problem that Pablo talked about, this idea of indeterminacy in the system, which is a mathematical problem. And I want to get a little bit more specific about what indeterminacy means that's relevant for us here in this workshop, in this presentation, which is that it is saying that given the available observations and the available statistical techniques, we are not able to infer the species interactions directly. And there are a few reasons for this. And I think as we better understand the reasons why our statistical techniques are currently failing us, we may open the door to develop better statistical techniques. And so one has to do, as I mentioned before, with this non-Gaussian species distribution. So even in this model, we find that we have a non-Gaussian distribution of species abundance. And so it's a problem particularly for covariance, but it affects the ability of any of these network reconstruction techniques. And so let's take the lesson from the previous section where we said that covariance structure may reveal species interactions. That's the hope. But what I find here is that the environmental stochasticity, so that's stochasticity and birth-death processes, combined with the drift term, so the propagation of stochasticity through the network, actually can generate covariance motifs that are not the real ones. And so just to talk a little bit more about methods before I get into the results, I use a range of network geometries, so ranging from 4 trophic levels, these 3 trophic level, and 2 trophic level. And I vary not only the network motif, but also the strength of interaction between species, which is this parameter D. And then the magnitude of the stochasticity, which is a parameter gamma. And so let's just look at the proportion of links detected. So just broadly, out of all of the links that exist, what proportion are detected? That's similar to the sensitivity that I was looking at in the previous section. And given this network architecture, random detection is a proportion of 0.5 because we have a network with 6 possible links and only 3 are actually connected. And so the proportion of the sensitivity we see does depend on gamma, but it's actually a stronger function of the interaction strength D. And unfortunately, the network architecture itself. So we see that some network architectures, in particular, this one with the 3 predators and 1 prey, is very poorly detected if we have strong interactions. It's so poorly detected actually that you detect none of the true interactions. So you only detect indirect interactions in this case. And so I'm going to focus on just 2 of these networks, and we're going to analyze them in more detail. What networks, if they're not reconstructing the true network very well, what networks are they reconstructing? And so take these 2 examples. And let's look at the number of true positives. So these are links that are detected in the association network that also exist in the true network. And so if the number of true positives is 3, we've reconstructed the network fully. And then the number of true positives 0 means we've detected only indirect interactions. And then we could have something in between. And so there's this star-shaped network, I'm just going to say the orange one on the left, that has random sensitivity actually. The sensitivity is 0.5, but there's actually non-random detection. So we're more likely to detect the true network than we have a better than random probability of detecting the true network, even if it's not great. We also have, on the other side, we have a network that has high sensitivity. So we're actually detecting a lot of the links, but we're not detecting the whole community. We're not detecting the whole network. And so I'm going to make this a little bit more detailed and say, what network are we detecting? And the network that we are detecting, I find, is so in this case, it's the true network. Here, what was really interesting to me was that when we have strong interactions, we're converging onto one particular network. And actually, in each case, we have a non-random probability of detecting a certain network. So we're differentially constructing a network architecture. And so this is telling me there's a relationship between the true network architecture and the network that we detect through association, even if it's not the true network. And so this is saying there's some information there, something interpretable, even if it's not exactly what we're looking for. And just to say, so I used actually 3 different metrics. So inverse correlation is maximum entropy, pairwise correlation, and then this algorithm limit that assumes that there is Lotka-Volterra predator-prey interaction. So you have a known model and you're just trying to detect the parameters of that known model. And so it's a kind of benchmark of this is the best known for this particular system. And we find that limits does perform better in terms of the sensitivity with strong interactions, but it's not necessarily performing any better in either the specificity or the sensitivity with weak interactions. But I'm going to turn to this question: why are we reconstructing particular networks, especially in cases like this where we find we're converging to one particular network? And what I realized was it truly is a property of the interaction matrix itself. And so here I'm plotting the largest eigenvalue of the interaction matrix in solid line and the symmetric part only of that interaction matrix as a function of the interaction strength for 3 different network modules. And what we find is that the largest eigenvalue of the symmetric part becomes positive. And this was termed by Newbert and Caswell in a 1997 paper as reactivity. And what this means is that the system is prone to transient growth. So if we look in species-species space, we find that although there is a stable coexistence point, stochastic perturbations can push the system onto long transients away from equilibrium, and the direction of those long transients is set by the eigenvalues of the symmetric matrix itself. And so if we look at the eigenvector components, what we find is that each of these cases of a reactive system, the most likely network by covariance has one species disconnected, and the species that's disconnected is the species where the long transients are most in the direction of that species. And so this one is this is a system that's prone to transients when it's perturbed, and that results in this specific network architecture through covariance. And so these are small networks. These are just 4 species. And our question was, could we generalize this to larger networks, and would we see similar results? And so I used the niche model, Williams and Martinez, 2000, published in Nature. And what this model does is it gives you a way to construct trophic interactions that seem to represent true trophic interactions. And so here I'm using 100 species networks. I've arranged them low to high trophic levels, and this is kind of the average true network. You see that these are asymmetric, right? Positive, high trophic level species are eating the low trophic level species. And so this is the average true network, but what we find is when we look at the correlation density from these association networks, they really are converging to a non-random network structure, especially when we have stronger interactions. But it doesn't really bear any resemblance to the true networks that we know from this niche model. And overall, we find that this is a reactive system. This niche model that seems to represent trophic networks well is also prone to these long transients with positive and actually very large positive value. Eigenvalues is the symmetric part of the interaction matrix. And the result of this is that when you have weak interaction strengths, you have high sensitivity, but fairly low specificity. So you detect a lot of the interactions, but you don't, but that's because the network is too complete. You're detecting a lot of the indirect interactions as well. But as the interaction strength increases and becomes more reactive, you lose the ability, you lose accuracy in network reconstruction. And so what we found here is that to some extent, whole networks of species interactions actually are detectable by these metrics that we use, covariance, correlation, and inverse covariance. I think one thing that was important here was that increased stochasticity did not decrease the detection of interactions. But we also found that there's non-random selection of network motifs that are not necessarily the true ecological networks. And so just for my conclusions overall, to begin to make progress towards this question, did we make progress towards this question? What is the relationship between species co-occurrence in space and time and the interactions between species? My answer is it depends on the structure and type of species interactions, which is a complicating factor, but also leads to some interpretability if we have information about the system. And we also find that abiotic factors actually have a more statistically detectable influence on species co-occurrence. And so the niche is really, probably unsurprisingly, what you're detecting when you look at species co-occurrence. And to the extent that biotic factors alter that, that's what you are able to infer. And so what do we learn about kind of future prospects? Where should we go from a mathematical perspective? One is here I was just using the distributions in space and time, but I think would be a lot of power in incorporating the rates of change, or another way to frame that would be entropy production, if we have that from time series. And then I think there's also would be power in incorporating environmental variables where available, and tools that look not just at biotic, but also abiotic factors. Yeah, and since I'm not here, I'm happy to have any questions now, or conversation comments, but I also put up my email because I'd be happy to talk over email as well. And then the papers that I discussed are on my website. which is that.
So thank you very much. I don't know who has any question apart from Pablo.
I was trying to connect to Zoom. Lara, can you hear me?
I can hear you. It's a little bit quiet, but go ahead.
We can also come.
I'll need to. Hi, Mara.
Nice
Hello.
talk.
Thank you.
Thank you.
Wait, that Paul is doing something here.
No, just to see you both.
You can actually stop sharing.
Okay.
So we can see you.
Ah, yes.
Oh, yeah.
Perfect. So two things. One is that it looked like stochasticity, because of you said about the transient dynamics. It looked like stochasticity will actually kill the May's 1972 conjecture, because probably the system will never be stable at all, no matter the number of species that you have, or it might be different, the condition in terms of the number of species and the stability of the system. What do you think about that?
Yeah, I think that's a good point. Yeah, so May's 1972 paper, they insert randomness in the interaction strengths. And this relates to, I think, Danielle's discussion of some of these different formulations for where do you put the different types of interactions. And so we had static interaction strength, but we had the stochasticity in a birth-death process. So I sort of demographic stochasticity, but the interaction strengths are static in some sense. And I think you're right that perhaps these long transients away from equilibrium can really alter. And I think it'd be really interesting to compare this stochastic differential equations framework we're looking at. It's a stochastic birth-death process to this sort of random matrix theory of these interaction variability and interaction strengths.
Yeah, I think.
I think another major limitation of that is the distribution of interaction strengths that May uses are not the known distribution of interaction strengths. I think interaction strength is difficult to measure, and so we don't necessarily know what that full distribution is, but it's probably not the Gaussian as we assumed. So I think that's another way where advances in random matrix theory might help us to improve our understanding there.
Yeah, the other thing is that, for example, Alesina, which improved on May's, but he also categorized the systems as there is a clear cut between predation, mutualism, and competition, and we know there is none because of these things interact. I mean, somehow there is some indeterminacy in what is predation, where predation ends and mutualism starts. So that's interesting. But the question I wanted to ask you is for the reconstruction of the intertidal network, it looks like you were able to pick up mostly positive interactions linked to habitat type and also to the presence of other species. And I wonder if that reflects the fact that it's easier to find a species for real than to know the reason why it's not present. So it's like a presence convey a higher kind of sense of certainty than the absence, because it might be absent because you miss it, because it never got there, because someone ate it, or maybe you are in the wrong place at the wrong time. But the presence is kind of stronger, isn't it? So to speak.
Yeah, absolutely. And so I think one of the limitations of our intertidal study is that we use presence absence only, and we don't use abundance information. And I forgot to say that, but one of the reasons why we don't use abundance information is because we were looking both at these like algae and crabs, and it's difficult to know how do you put crabs and algae in the same quantitative framework, right? Number of individuals is challenging when you're trying to observe it. And modeling is a different thing. And so, yeah, to go back to your question, I think it's a challenge. There's a sort of non-uniqueness of the system where you're only going to measure a discrete number of species in this grid, and you won't necessarily, that doesn't mean that you're capturing the full potential interactions. You're just seeing what was realized because of all of these other processes. Yeah, sorry, there was one other point, but we can.
Okay, thank you, Myra.
Yeah, thanks for your questions. Yeah, if anybody else, I'm happy if anybody else has questions too.
Everyone is happy. Okay, now maybe we just turn off now the recording, and we thank you again. So